dive right into the heart of the matter, the 6 stages of the evolution of gene editing, a journey that has revolutionized our understanding of life itself. Picture a world where we can edit the very code of life, the DNA. This isn't the realm of science fiction anymore, but a reality we're living in today, thanks to the advent of gene editing. Gene editing, in essence, is the ability to add, remove, or alter genetic material within an organism's genome. Journey back with me to the 1970s, when the foundations of this revolutionary technology were being laid. The early experiments in gene editing were not on humans, or even animals for that matter. They were on a humble bacterium, E. coli. These bacteria were the unsung heroes in the birth of recombinant DNA technology. Recombinant DNA technology, as the name suggests, involves recombining pieces of DNA. Scientists in the 1970s, armed with their microscopes and petri dishes, began to tinker with the DNA of these E. coli bacteria. They would cut and splice the DNA, creating new combinations, new possibilities. The result was a new form of life, a bacterium with a genome partially designed by human hands. This was the dawn of gene editing, the first stage in its evolution. These early experiments were not without their controversies. There were concerns, debates, and even a moratorium on this new technology. But the potential benefits far outweighed the risks. The door to a new era of biology had been opened and there was no closing it. The work of these early pioneers was just the beginning. Their experiments with E. coli paved the way for future advancements in gene editing. Each new discovery, each new technique, has built upon this foundational knowledge. Gradually, we've been able to refine our tools, increase our understanding, and expand our capabilities. Little did they know these early pioneers had set the stage for a revolution in biology. The second stage saw the advent of genetic engineering. This period, mostly in the 1980s and early 90s, was akin to a scientific renaissance, a veritable explosion of knowledge and exploration into the heart of life itself, our genes. Scientists began to unravel the complex tapestry of DNA, learning how to manipulate its threads to create something new. Among the first successful genetically engineered organisms was the humble tomato. But not just any tomato. This was the infamous Flavor Savr tomato. The Flavor Savr was no ordinary fruit. It was a marvel of genetic engineering, its DNA tweaked and adjusted to extend its shelf life. This meant it could stay ripe and fresh for longer, a feat that promised to revolutionize the way we eat. And while it may seem like a small step, it was a giant leap for the field of genetic engineering. The Flavor Savr tomato symbolized the immense possibilities that lay ahead. It was a testament to our growing mastery over the genetic code, a tangible example of science transforming something as everyday as a tomato into a beacon of progress. But as with all pioneers, the road was not always smooth. Despite its scientific success, the Flavor Savr tomato was a commercial failure. Yet it served an important role. It was a stepping stone, a learning curve, and a glimpse into the potential of genetic engineering. While the Flavor Savr tomato was a commercial failure, it paved the way for future developments. The third stage ushered in the era of gene therapy. Imagine a world where genetic diseases are no more a life sentence, but rather something that can be treated or even cured. That was the promise of gene therapy. The new millennium, in the year 2000, saw the first approved gene therapy treatment. This was a major milestone in the field of genetic engineering. The treatment, called Gendocine, was approved in China for the treatment of certain types of cancer. It works by introducing a functional gene into cells to compensate for an abnormal or missing one. A major leap indeed. But as with any groundbreaking technology, the path was not always smooth. The early years of gene therapy were marked by setbacks, including unfortunate patient deaths in clinical trials. These incidents served as a sobering reminder of the potential risks and ethical considerations involved in gene therapy. Despite these challenges, the field continued to grow and evolve. By 2010, approximately 500 gene therapy clinical trials had been conducted worldwide, a testament to the relentless pursuit of scientific knowledge and advancement. The last decade has seen an exponential increase in these trials. From 2010 to 2020, the number of gene therapy clinical trials nearly doubled, with over 900 trials conducted. This surge can be attributed to advancements in our understanding of the human genome, as well as improved techniques for gene delivery and editing. Today, gene therapy is not just a concept confined to the realms of science fiction. It's a real, tangible field of medicine with the potential to transform lives. From treating inherited retinal disorders to certain types of cancer, gene therapy has started to make its mark. And let's not forget about the potential to treat genetic diseases, which affect millions of people worldwide. Diseases like cystic fibrosis, Duchenne muscular dystrophy, and many others could be effectively treated or even cured through gene therapy. Despite initial setbacks, gene therapy continues to hold promise for curing genetic diseases. The fourth stage brought us the powerful tool of RNA interference. This groundbreaking discovery in the early 2000s forever changed the landscape of gene editing. It was a revolution in our understanding of gene regulation, and for their pioneering work, Andrew Fire and Craig Mello were awarded the Nobel Prize in Physiology or Medicine in 2006. RNA interference, or RNA, allows scientists to silence specific genes, turning off the production of proteins that may contribute to disease. This has opened up new avenues in the fight against genetic disorders, cancer, and even viral infections. It's like having a molecular switch to control the expression of our genes. As we delve deeper into the intricacies of our genetic blueprint, RNA interference has become an indispensable tool in the geneticist's toolbox. This is the age of RNA interference, a testament to human ingenuity and our relentless pursuit of knowledge. The fifth stage saw the revolution of CRISPR. CRISPR, or clustered regularly interspaced short palindromic repeats, was a game-changer in gene editing. Discovered in 2012, it was like unearthing a treasure trove for genetic engineers. This revolutionary technique was found in bacteria, which used it as a defense mechanism against viruses. Isn't that a twist? The very organisms we often fight against, holding the key to a whole new world of possibilities. Within a span of just a few years, the number of studies involving CRISPR skyrocketed. By the end of 2017, more than 20,000 CRISPR studies had been conducted worldwide. That's a staggering increase from just a handful of studies at its inception. These numbers only indicate the immense potential of this technology, but it's not just about quantity. The applications of CRISPR are as diverse as they are numerous. From treating genetic disorders like sickle cell anemia and cystic fibrosis, to enhancing crop resistance in agriculture, CRISPR has proved to be a versatile tool in our arsenal. It's even been used to create gene drives, which can spread specific genetic alterations through populations of species, potentially eradicating diseases like malaria. Indeed, CRISPR has been nothing short of revolutionary. It's akin to having a pair of molecular scissors that can precisely cut and paste DNA sequences, altering the very fabric of life. CRISPR has completely transformed the field of gene editing. And we're just getting started. The future holds even more promise as we continue to unlock the full potential of this extraordinary technology. Finally, we arrive at the sixth stage, the future of gene editing. As we gaze into the crystal ball of scientific advancement, we see the promise of emerging technologies like base editing and prime editing. Base editing, a technique designed to create precise genetic changes, could potentially correct around 67% of known genetic diseases. On the other hand, prime editing, the new kid on the gene editing block, holds the potential to correct almost 90% of genetic abnormalities. However, with such powerful tools at our fingertips, we face equally significant ethical considerations. How do we ensure these technologies are used responsibly and fairly? What are the implications of manipulating our genetic code? The future of gene editing holds untold possibilities and challenges. As we stand on the brink of this new frontier, one thing is certain: the journey of gene editing is far from over.